Forensic Science Lecture 10-2, DNA Typing with Tandem Repeats. Tandem repeats. Geneticists discovered that portions of the DNA contain sequences that are repeated numerous times within our DNA, our chromosomes. And this is known as tandem repeats. They act as fillers or spacers between the coding regions of the DNA. Although the origin of these are a mystery, forensic scientists, two forensic scientists, they offer a means of distinguishing one individual from another. There are numerous possibilities that exist for the number of times a sequence can repeat themselves. Sometimes it's 100 times, sometimes it's 1,000 times. All humans actually have the same types of repeats, but there's a large variation in the number of repeats. And so forensic scientists first began to apply this DNA technology to human identification in 1985. Until the mid-1990s, forensic communities aimed in efforts on characterizing repeats. And this is known as restriction fragment length polymorphisms, which are also known as RFLPs. With the RFLPs, what they do is they will cut the repeats from the DNA with a restriction enzyme. Restriction enzymes act like scissors. The typical sequence that they look for is anywhere from 15 to 35 bases long. And then the chromosome is cut into hundreds of fragments. Some of these fragments contain repeating sequences and some of them don't. The length differences with the RFLPs allow forensic scientists to distinguish from one person to another. After the restriction enzyme has cut the DNA, the fragments need to be sorted. And so they use a process known as electrophoresis. And this technique is similar to the thin layer chromatography that we talked about with the drug analysis, but it's specific to DNA. And this is accomplished by DNA fragments that are placed into a plate that's coated with a gel medium, typically agar. And then electricity is added to the gel, causing the fragments to move across the plate. Smaller fragments tend to move faster than the larger fragments. And so what this does is separates those fragments according to their size. The next step, after electrophoresis has been completed, the DNA is chemically treated to separate the strands, so the double helix. The fragments are then transferred to a nylon membrane, and this process is called a southern blotting. It's actually named for who developed it. But basically they are taking the plate and applying it to a nylon membrane, and it basically, like an ink blot, will transfer it over. Membrane is then treated with radioactive probes that contain complementary base pairs to the RFLs being identified. This process is called hybridization. And so this allows for the RFLs to be visualized, to actually be seen. And so once that process has been accomplished, then the nylon sheet is then placed against an X-ray film, and it's exposed to this X-ray for several days. The radioactive decay that is produced from the probe will strike the film. When you process the film, the bands appear where the probe stuck to the fragments in the nylon sheet. Typically, a pattern shows two bands, one from each chromosome, one from the mother, one from the father. With DNA typing, with using the RFLs. They compare DNA fragment patterns to known fragment patterns. So what they're looking for are matches between the band sets. Alone, with one set of band sets, it can't be used to individualize a stain to a suspect. However, a high degree of discrimination can be achieved, and they do this by using a number of different probes, and then comparing them and their combinations, so that increases the likelihood that it matches an individual. The first the RFLP DNA typing is actually the first scientifically accepted protocol in the U.S. that was used for characterizing DNA, but only for a short time. New technology has incorporated the polymerase chain reactions, or the PCR, to replace the RFLPs. The RFLP DNA typing actually was most notably brought to light with the President Clinton Monica Lewinsky case. This is what they used to prove that the semen stain was President Clinton's. This is the end of lecture 10-2. There's a quiz next class.